Good morning, everyone. I'm Helen Sjogren, and I work as a principal scientist at Ferring Pharmaceuticals. And today I'm going to talk about development of parenteral multidose products, mainly proteins. The outline of my talk, I will start a bit to introduce parenteral multidose product and then continue with different challenges with developing multidose product and especially challenges related to the addition of a preservative and how it affects your peptide and protein stability, incompatibilities with other excipients, chemical stability issues, and also effect on antimicrobial efficacy. Parenteral delivery, as you all know, is the dominant delivery route for biologicals, where injection pens are a patient-friendly product design and an option for delivering biologicals, at least if you have a reasonable dose. Injection pens are also the most commonly used multi-dose device, among others, because it gives the possibility for individualized dosing of the drug. And you find almost 200 different marketed injection pens from more than 80 different companies on the market today. But for all multi-dose products and injection pens, you need to add a preservative to your formulations. And that is what I'm going to talk about today. So if you look more general on the design of a biological product for parenteral use, except for your actual active ingredients, your protein, if you have a multidose product, which is almost a third of all protein products on the market today, contains then a preservative. Most products also contains a buffer, and 2/3 of them contains a surfactant to prevent surface absorption. Then on top of them, you can also find antioxidants or chelating agents. And again, most products contains one or more stabilisers or tonifiers. So this is roughly the content you can expect in a parenteral protein product. Not all parenteral protein products are well suited for multi-dose delivery. Antibodies, the largest group, are not, while hormones in many cases fit well to multi-dose delivery. In the other groups, it's a bit more varying, depending on dosing frequency and the amount you will need to give in one dose. There are more than 20 preservatives approved for parenteral use, but actually only a few of those are used today. So if you look again at products approved since 1995 in Europe, in liquid protein products, you only find 3 different preservatives: metacresol, phenol, and benzyl alcohol. And structurally, they are all rather similar, even though you need a quite different concentration of those to achieve sufficient preservation efficacy. Common for all preservatives is that they are having a destabilizing effect on most proteins and peptides. Insulin is a golden example where adding a preservative is actually good for both PK and stability, but most other proteins you see the opposite effect. So here I've shown that, illustrated that with 3 examples. So the first one is a Ferring protein, where when we have the formulation without a preservative, we have nice maintained protein purity up to 6 months storage, while when we add phenol to the same formulation, we lose almost 10% purity after 6 months accelerated storage. And with benzyl alcohol, it's even worse. There we have almost 20% loss of purity after 6 months. There are similar observations in literature. In this case, it's a graph showing the monomer concentration as a function of storage. And again, with adding different preservatives, you get a larger problem with aggregation over storage. Also, peptides show a similar example. So in this case, it's indirectly the amount of aggregate as a function of preservative concentration. And you see that additional preservative trigger your peptide to aggregate. All phenolic preservatives, like the 3 that are used in products, show similar phase behavior together with water, and they all have a so-called solubility gap. So that means if you look at phenol concentration in water as a function of temperature, and we can just focus on room temperature, which is roughly here. What you see is then at low concentration, you have a nice solubility of phenol, and you see the same, you have nice homogeneous one-phase solution at high phenol concentrations. But in the middle, you have a solubility gap. So you have a two-phase region. In drug products, you are always formulating down here. So in the phenol concentration below this two-phase border. But you need to remember this, it's important, the kinetics of dissolving phenol is important during product manufacturing. So you need to allow sufficient time for your preservative to dissolve before you continue formulating the other excipients and your protein. Except for preservatives, as I mentioned earlier, 2/3 of all protein products contains a surfactant, where polysorbates are the most commonly used surfactant, but you also find some with poloxamer. So it's likely that you will need to combine a surfactant with your preservative, and that can be really tricky. It's quite common that you end up with solutions looking like the the right-handed one here. They become turbid. The difference between these two samples is just a slightly higher concentration of metacresol and poloxamer in this sample than in this one, but it makes a huge difference on the phase behavior and the appearance of the solution. This problem with getting turbid solution, it's not specifically linked to the combination of metacresol and poloxamer. You see the same when you combine all these three preservatives with all the three commonly used or approved surfactant for parenteral use. And how can you understand this? What is this turbidity that you get sometimes and sometimes not? Well, it can be understood if you understand the clouding that you see for all PEG-based nonionic surfactants. So all PEG-based nonionic surfactants, so surfactants with an oxyethylene-based head group, show a temperature-dependent solubility, where increasing temperatures give a dehydration of the ethylene oxide groups. So you see actually a reverse solubility effect of increasing temperature. So the higher the temperature, the lower the solubility. And at some point, you get a turbid solution due to phase separation. And that was what we saw on the previous slide. And that is referred to as the cloud point of that system. The cloud point can be affected by additives. For example, sodium chloride, as illustrated here for polysorbate 20 and 80. But as you see, for this system, you are far above a reasonable temperature for a protein formulation. You will anyway never heat a protein formulation above 60 degrees. But if you add, instead of a salt, if you add a preservative, the reduction of cloud point is surprisingly large. And it's actually so large, so you end up below room temperature and even sometimes below refrigerated storage. And then you have problem. So here you see the cloud point. So the phase border between a clear one-phase system and a turbid two-phase system for polysorbate 20 as a function of phenol concentration. And what you can see here is that you get this U-shaped curve where a low surfactant concentration, here you can formulate rather freely at different preservative concentration, and the same at a high surfactants concentration. But in the middle, and that is actually the surfactant concentration that is most commonly used in protein products, then you have a problem. So if you look at the system at the phenol concentration that you normally need to get sufficient antimicrobial efficacy, you can formulate at low surfactant concentration and at high surfactant concentration. But in between, it's not possible to have a formulation, because then you will end up with a two-phase system, which is, of course, unacceptable. The limits here are also moved by other additives, proteins, stabilizers, buffer. So you need to understand this phase border for your specific formulation to know that you are sufficiently far away from cloud point of your system. Behavior is very similar for the three preservatives, phenol, metacresol, and benzyl alcohol, even though the preservative concentration when you see this clouding is different. The U-shaped curve is quite interesting. You see the same U-shape if you look at a pure surfactant water system and the effect of just increasing the temperature. So also here, a medium surfactant concentration give the lowest cloud point, while higher and lower surfactant concentration is actually easier to formulate. So this is clouding, and that is one problem with working with the combination, in this case of preservatives and surfactants. But there are other problems with preservative. And one is the interaction with other stabilizers. So most biologics contains one or more stabilizer or tonicity agents. These are the most commonly used ones. If you add a stabilizer to your preserved formulation, there is a risk that you get problems with chemical stability of the preservative, which is seen as yellow or brown coloration of the samples. And this effect is different for different preservatives. So while if you have a solution preservative phenol, adding a sodium salt is quite okay, but adding an amino acid like histidine or glycine will risk to cause brown solution. Benzyl alcohol, you see a similar pattern, even though what we have seen is for benzyl alcohol, adding, for example, glycine actually was the best option, while sodium salt, which was best for phenol, was the worst stabilizer to use with benzyl alcohol. And again, yet for metacresol, you see a different pattern. So the chemical stability of the preservatives is the kinetic of the degradation of the preservative is affected by stabilizer, but different depending on which preservative you use. And even though we understand the degradation routes of the preservative rather well, we do not know why different stabilizers affect the kinetic differently. The intention, as I mentioned in the beginning, to add the preservatives is to prevent microbial growth, and it's actually a must to demonstrate the effectiveness of your preservative according to all major pharmacopoeias. The test methods are rather much the same. You have your product solution, and you challenge it with 5 different microorganisms, you incubate it, you take samples from the incubated product solution with microbes after certain predefined time points. You dilute the samples in buffer through a serial dilution, and you grow the diluted solutions on plates and count the number of survivors. And from that, you can determine the reduction of live microorganisms after certain time points. And here you have predefined criteria from the different pharmacopoeias. Again, this preservation efficacy is affected by other excipients and also by the API itself. So the preservation efficacy is often reduced by the API. It's reduced if you increase pH. It's reduced if you add surfactant. You also often see a reduction if you add different buffers or stabilizers. But how large this reduction of preservation efficacy is depends on both which preservative you use and which stabilizer. And as for the chemical degradation, different preservatives show different effect when adding different stabilizers. So while you have a nice maintained preservation efficacy if you use a sugar together with phenol, and for benzyl alcohol, it's better to use an amino acids. So also here, the effect of different stabilizers is different. And to make it even more complicated, if you remember the chemical degradation, it's actually contradicting the preservation efficacy. So for chemical degradation and in a solution preserved phenol, it was best to use a sodium salt. But from a preservation efficacy point of view, a sodium salt was actually the worst selection that reduced the preservation efficacy to the largest extent. So it's a balance to develop a formulation that both shows a good chemical stability, but also maintained preservation efficacy. So to sum up, Multidose drugs require preservative. Around a third of all protein parenterals approved in EU since 1995 contains a preservative. Adding a preservative often reduces the stability of the protein itself, but the protein, along with other excipients, often reduce the preservation efficacy. And preservatives show different kinds of incompatibilities with different excipients. So one of the issues is that phenolic preservatives, they can phase separate together with surfactants. And therefore, it's important to understand the formulation phase behavior and map the cloud point for your specific system. And remember that both stabilizers, buffers, and proteins, and of course the surfactant concentration itself, affect the phase borders. Salt, sugars, and amino acids can also accelerate chemical degradation of preservatives. And especially the kinetics of this degradation is not yet fully understood and needs to be determined for each individual formulations. And with that, I would like to thank you for listening, and I would like to acknowledge Professor Marie Wahlgren from Lund University and Stefan Ulmerlund from Ensa Biotech, as well as Next Bioform, a research center that have financed the work that we have done together for mapping the phase border and cloud point of surfactant preservative systems. And finally, my colleagues from Ferring for helping preparing the other results I presented today, and you for listening. And thank you for letting me present here today.